Hello everyone and welcome to Pocket Size Bio with Dr. Casco. In today's video we're going to talk about Chapter 5 of Campbell's Biology. This is structure and function of large biological molecules. So let's get to it. So let's take a look at this first figure here, figure 5.1. Alcohol dehydrogenase, a protein that breaks down alcohol in the body, is shown here as a molecular model. The form of this protein that an individual possesses affects how well that person tolerates drinking alcohol. Proteins are one of the large classes of molecules or macromolecules. We're going to take a look at them in much more detail in the later sections. Okay, so one of the main things we're going to focus on in this chapter is what are the structures and functions of 4 important classes of biological molecules. Right? And these include carbohydrates, proteins, nucleic acids, and lipids. Now, lipids aren't macromolecules per se, but they're still incredibly important biological molecules. Okay. Also, we're going to talk about how monomers, basically the building blocks of these macromolecules, combine and chain up together to create these polymers, right? Or these very, very large chains of monomers. Okay. So let's move on to the first section, which is 5.1. Okay, just going to zoom in a little bit here. So macromolecules are polymers built from monomers. So large carbohydrates, right, proteins and nucleic acids, they're also known as macromolecules, large biological molecules. This doesn't include the lipids, like I just mentioned, right? So in general, they're called polymers, right? Because they're due to their size and their chain-like structure. Poly means many, meros means parts, so many parts, essentially. These polymers tend to have repeating units, right? And these repeating units are referred to as monomers. And you can think of monomers like little Legos stacking up and being built together, right, to create a larger structure. Okay, but each individual monomer can have a little function of its own, right? So synthesis and breakdown of polymers. All right, let me just zoom in and move down a little bit here. So there are many different macromolecules, but in essence, they have a similar process of building up, right? Of connecting together. And that process of creating macromolecules is referred to as polymerization. Polymerization, okay? And this process of polymerization is usually facilitated by enzymes, which is a group of proteins. Again, this is something we're going to see later in the chapter. Right? They're very special macromolecules, usually proteins, that they help speed up a chemical reaction. Okay? So, within this idea of polymerization, you have condensation reactions. And condensation reactions is basically any type of reaction where you have 2 molecules connecting together, and they're usually covalently bonded. And as a result of this covalent bond, you release a small molecule. Whenever that small molecule that is being released is water, it's referred to as a dehydration reaction. So one of the 2 molecules releases a hydroxyl group and the other releases a hydrogen. Okay? And the reverse of this reaction is referred to as hydrolysis. Hydro meaning water, lysis meaning to break. So it's basically the reverse of dehydration, synthesis. Let me just go a little bit down. So what happens? The bonds between monomers are broken down, and water is being attached to the resulting 2 subparts. And this happens a lot during the process of digestion, right? Enzymes help water come in and break a polymer into subunits, right? Into smaller parts. So they help speed up that reaction. And then those monomers would ultimately go into the bloodstream to cells to be used in a particular way. And what's interesting about these monomers is that they can still be used to create different structures depending on the cell that needs it needs to be used in, and the type of reaction or structure that it needs to be used in, right? So there's a lot of variety in that sense. Just to take a look at the visuals so you can see a representation of this, right? You have dehydration reactions. So here you have a short polymer consisting of 1, 2, 3 monomers. And then you have a fourth unlinked monomer that wants to join in. And in order for that to join in, if you see here, right, the hydrogen from the monomer number 3 and the hydroxyl group from the unlinked monomer would need to be removed together, right? And when they're removed together, you create that linkage there, that covalent bond that you see here, and a longer polymer is formed. The opposite, again, hydrolysis, you're breaking down that polymer via enzymatic reactions, and water would be inserted into that particular area that you want the reaction to occur in. Therefore producing having a hydrogen attached to the polymer and a OH group to the monomer. Okay. All right. As we're going to see later, there is a lot of diversity in macromolecules based on how these monomers interact and the sequence of these different monomers. You see this a lot with DNA and proteins. We're going to get into this a lot later in incredible detail, right? DNA is responsible for the hereditary information that is passed down from parent to offspring, right? And depending on how the sequence of DNA is, the nucleotides, you can have a sense of similarity between species, between different animal groups, and organisms in general. You can compare humans amongst other humans, right? And sequence DNA. Humans and other primates, you can see sequences, similarities and differences, or humans and fish, humans and invertebrates. So many different things can be compared just because of variations in the sequence of polymers, right? In the sequence of amino acids in the case of proteins, or in DNA in nucleotide order. Okay, so this is what the book refers to as polymers are made of a collection of monomers, right, that are sequenced in a particular manner. Okay, and it's a nice analogy in terms of alphabet and how we use the alphabet, where we have selected, letters in a particular sequence to create words, right? Analogous to that, right? Or as an analogy. Okay. Let's go to section number 2, 5.2, going into carbohydrates. So carbohydrates serve as fuel and building material. So this first group that we're going to talk about essentially includes sugars and polymers of sugars. They're referred to as a carbohydrate group. And when you talk about the simple sugars, this is called a monosaccharide. When you have two simple sugars together, this is referred to as a disaccharide, di meaning two. And when you have polysaccharides, this refers to as many, many simple sugars linked together, right? Poly meaning many. So focusing on monomers, right? You have the monosaccharide group. And generally, when you kind of want to know the chemical formula of this particular structure, you have this kind of formula, or this base formula to calculate for monosaccharides. It's basically CH2O, right? And you will have multiples of this ratio here. Monosaccharides generally have a carbonyl group, okay, and multiple OH groups, right? So you could see here, here's an example. So here you have a triose, which is a 3-carbon sugar, and you have this carbonyl group at the end of the chain. Anytime you have the carbonyl group at the end of the chain, you have aldoses and you have ketones if the carbonyl group is within the carbon structure. Okay. So the most common monosaccharide is glucose. All right, and it's an important molecule for the chemistry of life. And again, depending, you have this carbonyl group and you have a hydroxyl group for monosaccharides. Depending on the location, you can have an aldehyde sugar, which is referred to as an aldose, or a ketose sugar referred to as a ketose. Okay, Most sugars, their naming ends in oses. That can give you a hint as to identifying them, or whenever you read this word, you know already it's a sugar, most likely. Okay? And the name generally designates to the number of carbons, right? So, for example, 3 carbons is referred to as a triose, 5 carbons refers to a pentose, 6 carbons refers to a hexose also. Okay. There is also diversity. So you can get structural arrangement where you have structural isomers, right? But just by that structural arrangement, even though they have identical chemical structures, right, they might have different functions just because of their structure there. Okay, so just to take a look back at this the figures here. Again, you have aldoses, you have ketoses, ketone sugars, aldose sugars. Here you have an example of a 5-carbon sugar where the carbonyl group is at the end. Basically, this will turn into ribose, an incredibly important component of RNA. And when you have it in the within the carbon chain, you would have ribulose, an important intermediate in photosynthesis. Okay, again, always that same pentose. Hexoses, similar situation. Here you have glucose and galactose. They're both in a linear chain. You have a carbon chain here. They both have the carbonyl group at the end. But there's just a difference in their OH arrangement, right? So they're isomeric. Just by that difference, they're completely different sugars, right? You have glucose, galactose. Okay? And then you have another variant, so fructose, which is an energy source for organisms, where you have that, of course, the carbonyl group in the within the chain. You can also have sugars creating rings in aqueous solutions, right? Generally glucose and other small sugars, they'll tend to form rings because it's a more stable form. Here you can see an example. Again, you have a 6-carbon sugar in a linear chain, okay? And it can transit into this different form. Here you see it progressing this intermediate form until you get a ringed structure. And something interesting about nomenclature here. Here you'll see this often in chemical structures, right? Where each carbon is enumerated, right? It's designated a number here. So this is carbon number 1. Here you have number 2, number 3, and 4, and 5, and so forth, right? Depending on the amount of carbons. Depending on the amount of carbons that you use. Okay. So make sure you understand that. That's just an abbreviated ring structure, right? This is going to be very common convention that you're going to see throughout the book, okay? Okay, so let's continue on. 5.2.2, monosaccharides are a major nutrient source for cells. So you use, of course, sugar as your source for cell respiration to break down glucose to obtain ATP. But the carbon skeleton of sugars can also be used as raw material to produce other compounds. Okay, amino acids, fatty acids that we're going to take a look at also. So monosaccharides that are not being used immediately by the organism, they could be turned into a stored form, if I can say, right? And you basically can link them together to produce disaccharides. And whenever you have 2 monosaccharides linking together, that bond that holds them together is referred to as a glycosidic linkage. And you can have several variants, right? Depending on the monomers that you're using. You can have maltose. So maltose, you have glucose plus a glucose monomer. This is basically malt sugar, right? And it's involved in brewing beer. You have sucrose, is the most prevalent disaccharide. Okay, plants generally transport carbs from leaves to the roots in this form. You have lactose, that basically has glucose and galactose linked together. It's present in milk. Those people that are lactose intolerant generally have this condition, right? Lactose intolerance, right? Because they don't produce an enzyme called lactase that would help break down lactose. But you could use enzyme supplements to break it down, right? Symptoms of these lactose intolerant could include gases, having gas, and cramps from the bacteria that's in the intestines. Okay, and then these disaccharides could be broken down whenever you need that energy from these disaccharides, from these sugars. You can break them down to extract the monomers. Okay, here's a visual on the synthesis of maltose, right? For disaccharide synthesis, again, same idea of this dehydration reaction, right? You have a glucose and glucose. One of them participates in donating the OH group, and the other one a hydrogen atom, which will produce water. Okay, same situation for glucose and fructose. Okay, the carbons involved in that glycosidic linkage could vary, right? In this case, you have carbon 4 linked to carbon 1. In this case, you have carbon 2 linked to carbon 1, depending on the structure, right? So something just to keep in mind, that the number or the carbon number is important. Okay, moving on to a larger group of, I'm just going to move a little bit here. Moving on to a larger group of carbohydrates, you have the polysaccharides, and these are macromolecules that can go from 100 monomers to your 1,000 and more monosaccharides joined together by these same glycosidic linkages. Okay, and they can essentially serve as storage material, right? And they could either also be used for building material, depending on what you're focusing on. Okay, so structure and function of the polysaccharide is determined by the monosaccharides and how they're actually positioned in the glycosidic linkages. So the first thing we're going to talk about is storage, right? Polysaccharides and their function to store. So plants and animals are able to store many monosaccharides together when they're linked, right, together into long chains. And in this case, they're branch chains. Or you can store this sugar for later use, right? You always need energy, and you want to have a surplus of it that you can rely on to obtain energy when you're in need of it, right? So plants, they store monosaccharides in the form of starch, right? And starch granules you can find in the plant cell and plastids, right? And again, it's whenever you accumulate a surplus, you want it in that stored form. So it's basically a carbohydrate bank, right? Animals can potentially eat this and also assimilate, right? And you can find these basically as potato tubers, grains, and fruits. So it's a starch source for humans. The simplest starch is amylose, which is unbranched, or amylopectin, which is a branched form. And I'm going to get into the visuals in a little bit. Animals store polysaccharides in the form of glycogen, very similar to amylopectin, right, in that they're a branched structure. They're even more branched than amylopectin. They have free endings, so that means there's more area in which you can pick off individual monomers ready to be used for energy harvesting. So let's look in the actual visual here. In terms of plants, right, you have starch, right? In starch, you have these chains here. Now there's two variants. You have amylose, which is unbranched, so it's just that single chain. And then you have amylopectin, where you have the long chain, and you could have some side chains of sugar monomers branching out, right? So it's somewhat branched. If you look in animals, we have glycogen. Okay, and glycogen, they could be stored within muscle tissue, and you see that they're highly branched here, right? So there's many sub branches and there's many endings here for enzymatic activity to come in and pick out those ending monomers so we can use for immediate energy usage there. Okay, another form we're going to talk about in a little bit is cellulose, right? And now this is for plants, and cellulose is this polysaccharide that is used to create the plant cell wall. And this is a very, very linear, unbranched arrangement that enables hydrogen bonding to occur between adjacent cellulose structures. Okay? Oh, my apologies. So, as I was saying, you have these cellulose molecules that are unbranched, and because they're highly linear and unbranched, they can create hydrogen bonds, and these hydrogen bonds can form on parallel cellulose molecules that could hold them together, right? And that creates even more support structure for the overall plant cell wall to make it incredibly rigid, strong for support for the plant cell. Okay, we're going to take a look at this in this next little section. Okay. So structural polysaccharides, right? So structural polysaccharides is referring to structure, right? Support. And organisms, they build strong materials from structural polysaccharides, not just plants. We're going to take a look at other organisms. But for the majority, you have plants that produce cellulose, right? Cellulose is the major component of tough plant cell walls. And plants produce almost 10 to the 14 kilograms, approximately 100 billion tons in a year, right? It's the most abundant compound on Earth, right? And cellulose is a polymer that will link together the carbon 1 to carbon 4 glycosidic, creating glycosidic linkages between the monomers. Okay, there are some slight variations depending on the orientation of the glucose molecules, in such that you can have alpha glucose and beta glucose forms. Okay, and it's all about the orientation of the hydroxyl group. So you have starch, and starch, all the glucose are in alpha formation, right? So all the hydroxyl groups are facing one direction. You have cellulose that has beta formation, where you have them interchanging in the orientation of the hydroxyl group. So just to show you a visual here, let's say you have this carbon chain, right, of this 6-carbon compound of glucose. It can change into this orientation of alpha glucose, or it can change into this formation of beta glucose, where you have this kind of OH group in a different orientation. And then when it creates these rings and it creates these glycosidic linkages, the OH group can be facing either one direction, okay? In this case, you have alpha glucose monomers, or you can have this beta glucose formation where you have them interchanging, right? So basically one glucose is upside down from the other adjacent two, right? So there's this little zigzag formation, so to say. And because of that, just to go back to the notes here, you have a difference in 3D shape. Cellulose is never branched, so it's very, very fibrous, and thus creating that hydrogen bond. And because it creates that hydrogen bond, it's so sturdy and strong that it creates microfibrils, right? And it's a strong building material for plants. Humans can use this for paper production, for cotton, right? And so forth. So again, what's important is that creation, that development of hydrogen bonds, okay, between parallel strands of cellulose. Okay, enzymes can break alpha linkages, right? But these enzymes, they are not able to hydrolyze beta linkages. So that's how strong the resistance is to break apart for cellulose, right? There are very little organisms that are actually able to break down cellulose, okay, or in this beta formation. Most animals cannot, right? We eat, plant material. We have our salads. We have our fiber. It does help us with our digestive tract, right? It stimulates lining to produce mucus, and that mucus enables better movement for our waste material. So it's very healthy for our diet. It's not necessarily a nutrient, but it's very healthy for our diet, right? It's referred to as insoluble fiber. Those organisms that can digest, for example, like cows, now they don't produce the enzymes per se, but they have cellulose digestive prokaryotes, bacteria, and protists that are able to break down the cellulose, and then they could extract that nutritive content. Okay, termites can also break down cellulose. They have a very similar digestive mechanism. Fungi also can digest cellulose in the soil, and it helps recycle nutrients for a whole ecosystem, right? So that is cellulose. You have another structural form of polysaccharide, which is called chitin. Okay, And it's a carbohydrate used on exoskeletons for arthropods, right? For insects or spiders or crustaceans, you have this material that enables the formation of the outer skeleton. It is embedded also with a protein layer to help more resilience in the structure, or calcium carbonate for crustaceans specifically. Okay, fungi also has chitin, but it's a slightly different in terms of its structure, in the sense that it has a nitrogen attachment to it. So it's a little bit different than exoskeletons of arthropods. Okay, so this would be the chemical structure of chitin here. All right. Okay. So this finishes the first section of this chapter that is relatively long. I will create section number 2 in the days to come. Thank you for watching, and I'll see you in the next one.